A few years ago actually, I completed my PhD studying the bacteria that causes tuberculosis, which in fact is a slow-growing bacteria, is highly contagious, and it can be drug resistant. But now it's time to move on. And when microbiologists like myself move to study a new bacteria, we often stick or study a different bacteria within the same family or switch to something easier to grow in the lab. That way you can plan experiments from day to day, which leads to scientific discoveries and publications. But I've done the complete opposite of that. Allow me to introduce you to something that's much more slower than the bacteria that causes tuberculosis. It is contagious or transmissible, and it can be drug resistant. This is MGen, which is also known as Mycoplasma genitalium. And this bacteria causes sexually transmitted infections in humans, and unfortunately, it disproportionately affects marginalized groups. And in severe cases, when you don't treat this infection, it can lead to things like infertility or even cancer. And you might be asking, we've been wondering, why did I decide to study something so difficult in the lab? And that's because I'm trying to catch it before you or someone you know catches it, okay? And because this is a very important bacteria, it is important to be able to test this in the lab. And I'm going to talk about some ways on the challenges, we face when you try to diagnose this briefly. So one of the things is it's an extremely slow-growing bacteria because most bacteria will grow within a day. The bacteria that causes tuberculosis usually take within 6 to 8 weeks to grow. With MGen, it is much more slower. It could take up to 3 months to grow in the lab, and if you're lucky, maybe 2 months. And it is a fastidious bacteria. What that means is it is very picky. Imagine you have a toddler who is very picky with food. That's how this bacteria gets. It is very picky, and that can be quite frustrating. And also, it is resistant to many antibiotics, and that is because it lacks a cell wall that many bacteria have. Therefore, the drugs cannot target what's not present. And scientists really use what we call PCR machines to detect this in the lab. And I'm going to talk about how this works briefly. For a PCR machine to work, you need additional equipment like a computer or a gel dock to visualize your image. And this machine uses a variable temperature to amplify your DNA, and that's how you can know if a sample is positive or not. And usually the analysis will take about 2 to 3 hours if you're a very skilled technician. And it is quite expensive because you need to buy this machine, train the folks who are going to use it, and also the additional equipment you're going to be using for testing. What my research is doing, what we'll be doing in the lab, is to take a new technique called lamp assay. And basically what you need is just a heating block like that. You can literally get that on eBay. And what it does is it allows for colorimetric detection, which means you can visualize your results with your naked eye without the need for you to buy a new computer or something else. And it uses a constant temperature, unlike your PCR machines that uses various temperature for DNA amplification. And your results can be ready under an hour. That's quite quick compared to PCR. And it is cheap and easy to use, which means you can deploy this in under-resolved communities where they don't have equipment like PCR or facilities to carry out this kind of analysis. This is what a PCR result would look like when you conduct your analysis. Usually you'd have a graph, which is pretty complex to interpret, or you can have this kind of image here that also needs someone who is trained to interpret this. But in the lab, this is what a result looks like. You can see your tubes here, and you can see there's a color change, which you have red, which is the negative sample, and the yellow one, which is the positive sample. And this is 15 minutes of reaction. This is 60 minutes, which is an hour, which shows that this reaction actually works. So the relevance of this is to improve STI testing. And because time is of the essence, we know we can provide rapid detection of antimicrobial resistance, which would then prevent chronic stages of diseases and provide health equity for everyone. I would like to thank my professor and the lab for giving us resources to do this work, and also our gracious funders. And thank you all for listening.